I'm a behavioral physiologist, and what I want to do is to tell you a little bit about the work that my field has been doing, trying to understand how the brain forms a representation of space. And it's going to basically be a very, very kind of fast trip through some of the history of this, but I will try and tell you a little bit about what we have been doing, looking at more complex and slightly more naturalistic space than people have looked at until now. And I'm not sure if this has much to do with cognitive architecture as people think about it here, but I'm hoping that it might generate some ideas anyway. So I want to talk about spatial cognition as a kind of a model system. It's a form of knowledge, and the organism can use this knowledge to behave adaptively, and hopefully with artificial intelligence we can learn something about how the natural brain has solved some of these problems. So one of the things that we know from decades of kind of research behaviorally now is that when an animal is solving a spatial task, there are two systems that operate more or less in competition. And there are various names for these systems, but recently people have started talking about model-based and model-free behavior. And a really nice kind of illustration of this, is there a pointer?
Oh.
Okay.
Yeah.
Oh, okay.
There is a button.
Right, great. So a really nice illustration of this was a very old experiment on a plus maze where rats were started from one of these two start arms, and they were either trained to always turn right, so that sometimes they'd go this way and sometimes they'd go this way, or they were trained to always go to the east or the west, so sometimes they would have to turn right and sometimes they would have to turn left. So when they were trained on always making the same body turn, that was called kind of response learning, and when they were trained to go to the same place, that was called place learning. And what Tolman and his colleagues found is that they were very much faster at doing the place version of this task. So they learned it much more quickly with fewer errors. But we know that the brain can kind of use either of these forms, and it can switch from one to the other. So if you train a rat for a long period of time, it will switch from model-based to model-free learning. It'll develop automatic behaviors. So we know, at least just from studying behavior, that there are two brain systems for spatial navigation. Now we think that the model-based, place-based behavior is kind of sustained by the hippocampal system. So here is the kind of hippocampal system in three different species. The rat is the animal that we mostly study at a physiological level, and the hippocampus is this yellow structure here. It's very large relative to the rest of the rat brain. And in primates, it gets tucked down in the medial temporal lobe, and it's relatively much smaller because of all of the cortex that humans and monkeys have, all of the neocortex. But we still have a hippocampus, and it looks pretty much like the rat hippocampus and seems to do all of the same things. So the reason that we kind of started to think that this might be the site of model-based spatial learning is this very kind of seminal discovery by O'Keefe, who was recording from the dorsal part of the hippocampus in the 1960s. And people were interested in this area because it seemed to be involved in memory of some sort. And he used a technique that had recently been developed by Jim Ronk to record from freely moving animals, which had never been done before. And what he did was that if you have an electrode implanted in dorsal hippocampus and you collect the action potentials that electrode can see, and you plot them on the path of the rat as seen by a camera overhead. So if you trace where the rat has been and then kind of match up the action potentials to where the rat was, the thing that O'Keefe discovered and has been replicated many, many times since is that the action potentials from one hippocampal neuron all occur in the same place in the environment. So it was a very kind of remarkable finding. I've got a video of it. I'm not sure how well it's going to work, but we'll see. So this is a video of the rat walking, at least it will be, rat walking around. And if you listen carefully, you can hear the spikes, which you can also see on the oscilloscope trace over here. So it's very quiet at the moment. Can you hear that? It's a bit faint. But basically, whenever the rat goes into this region, you see a lot of activity on the oscilloscope, and you can hear the sound of the place cell. So this area here, this rat doesn't like to go there. So This area here, you can hear that, we call this the place field. And the question that has intrigued many of us in the field is, how does that cell know where the rat is? How does that cell know that it should start firing there? So this box is pretty boring. It's very symmetrical. But the cell knows to fire in this part of it and not in this part of it or this part of it. So it can break the symmetry of the box. I can also tell you that if you turned out the lights, that cell would still fire in that same place. You could take the rat out of the box, turn the box around and pop the rat back in, and now it will still fire in that place, even though that's physically a different part of the box. So the cell isn't relying on the smell of the box or anything about the local environment. It's something about the place. It's a very, very abstract concept. And it's very kind of intriguing to think about how that cell knows where to fire. So over the kind of years since, we've found out a lot of things, which I haven't got time to tell you in detail. One of the things that was established very early on is that they're multimodal. So as I said, they'll still fire if you turn out the lights. But if you have a visual cue in the environment, you take the rat out, you move the visual cue and put the rat back, now the place field will rotate its activity. So they are able to use vision. So when vision is present, they will use it, but when it's not present, they can use other things. So they can use a variety of different sorts of sensory information. They're omnidirectional, so it doesn't matter whether the rat runs this way through the place or this way, where the visual, panorama is completely different, that cell will still fire there. So it's not dependent on the visual snapshot that the animal can see. It's something much more abstract than that. I should say that that omnidirectionality is if the animal can move freely in all directions. It gets slightly more complicated if the rat is constrained to run backwards and forwards. Now the cells will typically only fire when the rat runs in one direction for reasons that I won't go into. The other thing is that the representation is non-topographic. So the first thing that you think of when you encounter these cells is that they're a bit like visual cortical neurons, and that there would be a map that's very much like the visual map of space, where neighboring neurons encode neighboring parts of space, but that's not the case. So if you have an electrode sitting in the brain, and there are two neurons being detected by this electrode, so they're next to each other. Typically, their place fields, the place where the cell fires, will be in completely different places. So there's no connection between where the cells are in the brain and where they fire in the environment. It's a non-topographic representation. The other thing is, and it's sort of related, I guess, is that the cells are randomly reused in different environments. So if you've got one environment, and let's say you've got this set of place cells active, this cell fires over here, this one down here, and this one up here. If you take the rat out and take it to a new room and put it in a new apparatus, some of the same cells will be active. So the blue and the green cell are still active in this environment, but in different relative locations. So they're not encoding the specifics of the geometry. A new cell has become active that wasn't active in this environment, and this one has stopped firing. And again, if you go into a third environment, it's different again. The Moses are the people who have done this experiment to the greatest extent. So they're recorded in 11 different rooms, and they found that, as far as I can tell, it's a completely random representation. So there's no connection, more or less, between the representation in these different environments. And then finally, there seems to be a population code for context. So what I mean by context is the non-spatial attributes of the environment that serve to distinguish one environment from another. So for example, if you record, so let's just look up here at the moment. If you record in a black box or a white box that's in the same place, so it's at exactly the same dimensions, everything about the box is the same except that it's either black or it's white, then you will get some place cells which distinguish those two conditions, and they'll fire differently. So this cell is firing in the northeast corner when it's black and in the northwest corner when it's white. So although the cell is encoding something spatial about the environment, it's using the non-spatial information to modulate its spatial activity. And we call this context information. So it's not specifically spatial, but it characterizes the space. So you can make this more complicated. You can say, as well as making the box black or white, we'll now give it an odor. So we'll make it smell of lemon or smell of vanilla. So now there's 4 different ways that this box can be. It can be black and lemon. It can be black and vanilla. It can be white or lemon. It can be white or vanilla. So this place cell fires whenever the box smells of lemon and doesn't fire when it smells of vanilla. So it's responding to odor. When it's lemon, as we saw, it remaps, so it fires differently. This is a simultaneously recorded cell. So this was active on the same electrode as this one. This one is not distinguishing the black from the white if the box is lemon, but it does distinguish the black from the white if the box is vanilla. So it's getting information both about the color and about the odor, and not only that, but its response to the odor is modulated by the color that's present. So it's integrating the contextual inputs that are coming in. So these cells are integrating their sensory information, and also they're behaving independently. So these 4 cells, all simultaneously recorded, have all done different things. So if you're a rat and you're saying, I wonder which box I'm in, you have to look not just at one place cell, but at lots of them. Because if you just looked at this cell, you would know whether you were in the white lemon box or whether you weren't in the white lemon box, but that's all you would know. To know exactly which box you're in, you need to sample a lot of cells. So it's a population code. So the full picture of where you are is distributed across the population of place cells. So, the brain is forming a very kind of flexible representation that it's able to kind of assemble from the incoming sensory information. Now, there's another kind of important phenomenon that's been found by place cells, and that is that although we typically record their activity as the animal is kind of walking around, when the animal is stopped or when it's sleeping, we also see place cells active, and they're active in a slightly different way. So when they're walking around, trotting along, if you record the local field potential, you see theta rhythm, which is this very large-scale sinusoidal oscillation, and you see place cells active, and they're active in bursts. So they typically fire these bursts, which are somewhat synchronized with theta, except not quite, but I won't go into that. So as the rat's walking through this space, it walks first through the place field of the red cell, so that cell fires, and then through the place cell of the blue field. And there's a little overlap here where these cells are kind of both active together. Now, if you record a bit later on while the rat is sleeping, then you see these same cells active, and they're active in temporally the same order. So in fact, there's a little period of time here where both cells are active, but the red cell fires slightly before the blue one. And we see that same order of events happening in a much compressed time scale. So whereas it took maybe 1 second for the rat to walk across this distance, in 100 milliseconds, you get this very high scale replay of these events. And we now think, for various reasons, that what's happening during that replay is that the system is going over what just happened and maybe using that to consolidate the memory, or maybe even using that for some kind of spatial inference. We don't know what it's for yet. Here's a nice video of replay that was made by David Foster, who's done a lot of really elegant work on this. So what he's done is he's recording during behavior, and then he's recording when the rat is sitting. It's not sleeping at this point, it's just sort of sitting. And what he does is he records place cells, large numbers of them, and because he knows where the place fields are in that environment, he can use which cells are active to work out where the rat was. So he uses a Bayesian decoding procedure to kind of retrospectively work out where the rat must have been given the cells that were active. So it's Bayesian decoding. And the Bayesian decoding is shown by the red kind of, I'll use this, these red blobs. So when the rat is walking around, so behavioral decoding, you can see that the red is pretty much at the place where the rat actually is. So it follows the rat around. So in other words, as the rat's walking, the cells that are active are the ones whose place fields are there. But when it's sitting still, thinking, let me just play that again. At least we think it's thinking. Okay, so behavioral decoding, the activity is kind of at the place where the rat is. So it's following the rat around. When the rat's paused, now you can still see activity, and it seems to be sort of coordinated in it that it seems to follow these routes. So it follows these kind of lines through space. And it's almost as if the rat is thinking about where it's going to go next, or thinking about where the goal is. And there are various labs looking at this phenomenon and trying to work out exactly what this information kind of contains. But certainly it seems to be evidence that the system is able to take things that have happened in the past and to regenerate these independently of experience. So something like an internal simulation of space. So what does that tell us about cognitive architecture? Well, so it seems like the place cells are forming an internal map of space. And this was quite an important discovery at the time because back in the 1960s, we didn't really think that the brain could do this, or they didn't think that back then. Didn't think that the brain had these internal representations. Everything was explained in terms of inputs pretty directly connected to outputs. So the idea that there was an internal representation was not well accepted. And it took a very, very long time, and John O'Keefe spent many years trying to kind of convince people, and it did take a long time. They reveal latent learning. So a place cell will form place fields no matter what the rat's doing in the environment, so long as it's able to walk around. So the rat doesn't have to have a task. Normally they're foraging, so we kind of will throw rice in to encourage the rat to walk around, but they're not navigating as such, they're just kind of wandering around. So these cells are forming their representation independently of the goal, the spatial goals of the rat, so it's latent learning. They may be evidence for memory. So the existence of a place field isn't necessarily evidence for memory. So they could be hardwired, but we do think that they probably are, to some extent, learned. So it seems that when a rat goes into a new environment, a hippocampal cell has the capacity to fire, and when it fires, something happens, some kind of plasticity, such that in future, when the rat goes back to that place, the cell will fire there again. The reason we think that is that you can artificially stimulate cells to fire in a place, and that cell will develop a place field there. So they're a mixture of hardwiring, which gives them a predisposition to fire, and plasticity and learning and memory, which makes them subsequently fire in that place. Now, that's not the same as being memory for events or the episodic memory that we think the hippocampus also does, but it does seem to be evidence that there's kind of memory in formation of this map. So that's quite interesting. They may be involved in the processing of novelty and familiarity. So when the animal goes back into that place, the place fields that formed, and it takes a few minutes for them to form, but once they form, they're present for many days or weeks afterwards. So they're very, very long lasting. And we know that the hippocampus is necessary for familiarity, so for recognition of environment. And we also know that if a rat has a lesion to its hippocampus, it will constantly be exploring. So it will always be walking around actively as if it doesn't know where it is. So for all of these various reasons, we think that the place cell system is forming a representation of space such that when the rat goes back into that place, it uses the sensory information coming in, compares that with the stored representation of that place, and decides whether it's the same place or whether it's a new place. If it's a new place, the place cells will remap, a new kind of representation of the environment will form, and new kind of associations will form. If it's the same place, then the rat will kind of know what to expect there. So that's a very important faculty, because often when you go back into a place, there will be things that will be different about it. So the furniture may have been moved around, the lights may have been switched off, someone may have repainted the walls or something. So the brain needs to be able to decide, is this a place I already know that's changed a bit, or is this a new place? So the deciding it's the same place is called pattern completion. So you take the information that's coming in and say, yes, this matches my stored representation, this is a familiar place. And the deciding that it's a new place is called pattern separation. And we think that the hippocampus is doing both of these things. It's doing pattern completion and it's doing pattern separation. And you probably know more about the models of this than I do, but there are strong arguments for thinking that the dentate gyrus, which is the first kind of way station into the hippocampus, is doing pattern separation, and the CA3 kind of network, which is the next one down, which is very interconnected, is doing kind of pattern completion. And it's all in service of deciding kind of whether you've been there before or not. And then as I showed you, there's this replay kind of phenomenon which might be used for something that we don't know. But there is a lot of evidence that the hippocampus is involved in memory consolidation. So that may be to do with kind of going over the information that's just been acquired and selecting some of it for storage for long-term use or something like that. And it may also be that these representations can subsequently be acted on to do things like spatial inference. So you've made your way around an environment, you now have kind of encountered the structure, and maybe when you kind of simulate that subsequently, you could do things like work out what are the shortest paths through this space and so Okay, so that was place cells. So about 20 years after place cells, it took a long time. This field has kind of moved quite slowly in some ways. So the question was, how do place cells know where the rat is? And one of the obvious ways to ask this question is to record from areas of the brain that connect to hippocampus. So people started recording in the regions of cortex that feed information into the hippocampal system. And one of the people who did this was Jim Ronk, who was one of the kind of principal people who developed this method for recording in freely moving animals. And he discovered that if you record in a kind of typical apparatus, cylindrical apparatus, from post-subiculum, which is kind of up here, then there are some cells which fire all over the place. So they don't have place fields, but they fire only when the rat faces in a particular direction. So it might be only when it faces over here, for example. So the way to kind of show the activity of these cells is to make a polar plot of firing rate against head direction. And one of these cells will fire at a very high rate when the rat faces southwest, let's say, and almost zero everywhere else. So it's a very, very kind of highly tuned signal. And if you record several of these cells, then they will have different kind of firing directions. And altogether, they seem to have the capacity to encode all 360-degree directions of space. So he called these head direction cells and proposed that what these cells are a sort of a compass system in the brain, and what they do is tell the place system which way the rat is facing. So shortly after he discovered these, a postdoc came to the lab, Jeff Taube. This is how they both look kind of in the last few years. They didn't look like that back then. So Jeff was quite young, and he kind of came to Ronk's lab, and Ronk said, look, I've found these weird cells. Why don't you kind of record these and see what they do? And so Taube spent the next kind of 30 years uncovering the network of these head direction cells. And he almost single-handedly, not quite, so and other people have contributed, but Taube has done the bulk of the work. And he's found some very kind of interesting things. So they're found not just in post-subiculum, but in fact in all sorts of areas. And we don't know why the brain needs so many head direction areas. But some of these areas are subcortical, so particularly the anterior thalamus and also the lateral mammillary nuclei. And some of them are cortical, particularly the post-subiculum where they were first found, retrosplenial cortex, and medial entorhinal cortex. So there are kind of cortical and subcortical regions. And Taube's argument, which seems pretty well founded in the data, is that the subcortical regions are collecting ascending information that's come from the vestibular system, and the motor systems have come up through the brain stem and are kind of ending up in the thalamus. And the cortical head direction areas are collecting information about the environment, so which landmarks you can see and so on. And again, it's feeding it to the thalamus. And the anterior thalamus may be the kind of place where this all kind of comes together. The second thing is that the head direction signal is local. So the rat doesn't use kind of magnetic cues or a sun compass or anything like that. Each room has its own directional kind of orientation pretty much, although there's a kind of an exception to that, which I'll tell you about. So the representation is pretty local, and it's kind of organized by the landmarks that are present. So the system is using mostly visual information to know kind of which way the rat is facing. But they can also integrate self-motion information. So this was shown very nicely by this experiment by Taube quite a long time ago, where he had two rooms, so two kind of chambers that the rat had to kind of walk between, and a corridor that connected them. So what he did was he recorded a head direction cell in one of these rooms, and then he recorded in the corridor as the rat walked out of the corridor and into the other room. And in both of these rooms, he had a cue card, but in the cylindrical room, the cue card was in the east, and in the rectangular room, the cue card was in the north. And what he found was that provided the rat remained oriented, and so you didn't take it out and you didn't disorient it by turning it around and so on, then a given head direction cell would maintain a coherent orientation between these environments. So when it walked into the corridor, the cell still fired when the rat faced north, even around here where it couldn't see this room anymore. And when it went into the rectangle, it still fired north. So provided the rat remained oriented, there was a coherent representation of direction between these rooms. So the only way that this cell could have known that this was north is if something about the movement cues that the animal made, probably the vestibular system and the kind of locomotor cues and the motor efference copy and so on, carried its kind of north signal through these corridors. Now, if you took the rat out and then just placed it randomly back in one of these apparatuses, then the cell would fire in the appropriate direction. It would still fire north. So once the rat had been in that apparatus and acquired the directional representation for that room, now the landmarks were enough to reinstantiate that representation. And we know now that that happens very quickly. It takes seconds or minutes. So when you go to a new place, you very quickly map the landmarks that you can see onto your internal sense of direction. And once that's happened, it's a very, very robust signal, and it's very difficult to reorganize it, which is a very kind of important finding when it comes to understanding how we remain oriented in complex spaces. So then the final thing about the head direction cells is that they act coherently. So if you have more than one head direction cell, whatever you do, whatever manipulation you make to the environment or the rat or whatever that rotates some of the cells will rotate others by the same amount. So the relative angles between these cells is always the same. So it's a very, very coherent signal. The head direction cells don't act independently in the way that place cells do. So yes, so head direction cells, so how do they remain coherent? The dominant model, possibly the only model for how this happens, is the ring attractor model, and we've encountered that already this morning. So a ring attractor basically is an idealized kind of connection of neurons where the active neuron is strongly exciting its neighbors and inhibiting the ones that are further away, such that activity is only ever localized to a small section of the network, which represents the direction that the rat is facing. So if the rat turns its head, then a signal will move activity around that network, and now these cells will capture the activity and they'll suppress activity everywhere else. So, the network only ever has one place that's active at a time, so you only have one directional orientation representation active. In this particular model, we think that there's some kind of visual inputs that, like landmarks or something, that help drive the activity in a particular ring, which is then kind of stabilized by the attractor dynamics. So that's why we think that the neurons are always coherent, because they're talking to each other as well as talking to the sensory inputs that are coming in. And just as an aside, I'll say that this model has been extended very elegantly to 2 dimensions, possibly to explain how place cells can also update their activity even when sensory information has been temporarily suspended. Now we have very recent evidence, which we've not published yet, that the attractor head direction system is a bit more complex than we thought. And this was a completely unexpected finding. But what we were doing is we were recording in 2 boxes. So this is kind of 2 rectangular boxes connected by a doorway. And what I wanted to do is to kind of look a little bit more at these context cues that I told you about before, where one box is centered with lemon and one of them is centered with vanilla. And there's a cue card at the west end of one box and the east end of the other box. And the idea was simply to see whether head direction cells can use the lemon or the vanilla. Yeah, I could take some questions.
Yeah. Oh, it's coming back.
Hi. I have 2 questions, but let me take the first one. You're measuring cells in mice. How do you know the mice have the same head each time? Right? It could be that the representations are different and they're not a fixed effect, they're a variable effect. So you're getting that, some mice might use smell, some might use space, some might use a different space. How do you know you're getting the same type of mice on that level even?
Well, I mean, the cells in all of the animals seem to use multimodal information. But it's true that maybe some animals attend to different cues than other animals do. So there's often a little bit of variability. For example, if you do a remapping experiment and you make the box go from black to white, you'll sometimes get a rat that just doesn't care about the difference. So there are some individual variations, but there are some commonalities too. One is that place cells and head direction cells use vision a lot. even though the vision in rats and mice is quite poor, it's nevertheless very important for the spatial system. And I think that's because visual information is so spatially precise, so it's very useful for localization.
Okay.
Okay, I'm not touching anything. We've got a lemon box and a vanilla box. And the idea was simply to see whether a head direction cell could use the olfactory information to know which box it was in, and therefore that it was in, whether this was the west or the east card that it could see. So should it fire this way or this way? So basically, can head direction cells process context information in the way that place cells can? And this was meant to be a new task for my new postdoc, just to get him kind of used to recording. And he came back and he said, something really weird is happening because I'm recording these supposedly head direction cells in retrosplenial cortex, but they have 2 tuning curves instead of 1. So if I do a polar plot, the cell fires north and south. And this seems kind of weird. So what he did was he looked at the firing as a function of which compartment the rat was in at a time, and he found that if you separate it out by compartments, in one of these compartments the cell fires when the rat faces north, and in the other one it fires when the rat faces south. So when the rat stepped through the doorway, this head direction cell kind of rotated. So that in itself is not that surprising necessarily, because the visual cues have also rotated, so everything is kind of symmetrical. It might just be that the cells didn't kind of receive any olfactory information. That was fine. But he also found other cells at the same time that did know the difference between the boxes. So here is a simultaneously recorded cell that always fired when the rat faced south, no matter which compartment it was in. So at the same time, there was a cell that looks like a head direction cell that rotated its activity when the rat stepped through the doorway, and another cell that didn't rotate its activity. So these cells have separated in their directional tuning. That's not supposed to happen with a ring attractor. So we're trying to kind of understand what's going on. And the kind of current thinking is that this is a system that's very close to visual cortex, whose job is to mediate between the landmarks that are coming in through the visual system and the head direction signal that's coming up from the anterior thalamus. And the idea is that these cells have to decide, is this a stable landmark with respect to the head direction signal or not? And if it's a stable landmark, then it will connect to the ring attractor. So here are 2 kind of hypothetical flip cells, one that responds to this landmark and one that responds to this landmark. They're both talking to the ring attractor. So when the rat faces northwest, sees this landmark, that drives the activity in that attractor and so on. Now, this landmark moves. So now you're over here. This landmark has moved. This flip cell is still, from this landmark, is still talking to the same part of the ring attractor, but this flip cell is now talking to a different part of the ring attractor. That's an unstable relationship, and so that should disconnect. So that's our kind of current hypothesis that we're trying to test, that these flip cells, which are these cells that rotate, these are not strictly speaking head direction cells, even though they look like it. And they talk to head direction cells, but they also kind of talk to the landmarks.
So you assume that they fire based on visual input? What happens in the dark?
So in the dark, they still fire. So it's not just visual. So you know, I mean sensory input, really. So at least in a familiar environment. So we haven't tried in a novel environment with the lights off. So we've started to model this. I'm not going to talk you through this, but just to say that we are in the process of trying to kind of think how this could actually work. And we've been kind of modeling plasticity between the visual system and the ring attractor, and that's kind of ongoing at the moment. So, so what have we learned from head direction cells that kind of adds to our kind of cognitive architecture? So these cells are proof of a metric signal. So place cells, although we think they're to do with place, there's nothing about a place cell firing field that proves that the system is spatial, because the cell fires in a particular place, but it could just be a conjunction of cues. The thing about head direction cells is that they encode direction no matter whether the rat is looking at the landmark or whether it was facing the other way, you turned out the lights and it rotated 180 degrees, and now it's using self-motion information to update its firing. So the signal that the head direction cells generate is integrating these two sensory sources of information whose only commonality is the metric property of direction. So one of them is a static cue and one of them is a dynamic cue, but they both signal direction. So this is the first time we've really kind of seen in the brain a truly metric signal. They reveal formation of a supramodal representation. So by the same argument, it doesn't matter whether the rat sees the landmark or just works out which way it's firing based on self-motion cues, path integration. So the head direction cell is abstracting direction from the sensory information. It's a supramodal representation. It suggests this new type of processing, attractor dynamics, which had been kind of postulated previously but not seen in the brain. And I should say at this point that we've not proved yet that there is attractor dynamics anywhere in the brain. So all of our evidence is circumstantial, and it's a bit hard to kind of falsify the attractor hypothesis, if I'm honest. But it's by far and away the most successful model in explaining some of the kind of activity that we see. So that was head direction cells. Then 15 years after head direction cells, a new discovery was made, and this is the grid cells. And again, the motivation was to look at the cortex that projects to hippocampus and try and understand how the place cells get their information. And the Moser lab were recording in the part of the neocortex that projects most strongly to dorsal hippocampus. And the anatomist Menno Witter, who's studied these pathways in a great detail, said, what you should do is look in the dorsomedial part of the entorhinal cortex, which is this bit here. So they started putting electrodes in there, and this is work that was done by this couple here, Torkel Hafting and Marianne Fern. And what they found is that there are neurons in this area of the brain that produce what look like place fields. So if you record in a small box, then you see these kind of focal patches of firing, just like you see with place cells. But instead of there being just one place where the cell fires, there are multiple places. And if the box is big enough, you see many of these patches of firing. And the really remarkable thing about them is that they have a very, very constant distance between them. And they're aligned in a very constant direction. So every time you put the rat back into this box, this particular neuron from dorsomedial entorhinal cortex will fire in these places. And these places are characterized by being 30 centimeters apart or whatever. So this is another metric signal. It's a metric signal for distance. And it's also expressing something about direction, because these kind of lines are always the same for all of the neurons. So with some caveats, all of the neurons that are simultaneously recorded. The other thing they found is that these distances vary. So as you go deeper and deeper, more and more ventrally into the entorhinal cortex, the distances between these firing fields get larger and larger. So it looks like the job of these cells is to do some kind of odometry, some kind of distance measuring at multiple scales. And that gives all sorts of interesting theoretical possibilities for spatial computations. So not long after grid cells were published, the people in my lab were very, very excited by this and said, can we record these? Can we record these? And I said, okay, you know. And I knew that they were hard to find, and they were hard to find, but Caswell Barry managed to find them, and he did an experiment that was motivated by an experiment that Neil Burgess had done with John O'Keefe some years previously. So Neil and I were co-supervising Caswell, and they said, can we do this experiment? And I said, this will never work, but yeah, all right, go ahead. And so what the experiment was is to see what would happen if you recorded a grid cell in an environment and then changed the shape of the environment so that you kind of compressed it in one or other dimension. And based on the work that Neil Burgess and John O'Keefe had done with place cells, there was reason to think that that would shrink or expand the spatial representation, because that's what place cells do. But on the other hand, these grid cells seem to have their own internal kind of metric for space, and it seemed to be kind of independent of environment, so it shouldn't scale. So the question was, what would happen? And what they found was that, surprisingly, they do scale. So if you take a rat that's familiar with a big square box and then put it in exactly the same box, but where the east and west walls have been slightly moved together, then the grid slightly compresses. And it's easier to see with this autocorrelation kind of pattern. So these blobs are getting slightly closer together, only in a dimension where the walls have moved closer together. And if you do it the other way, then they squeeze together in that dimension. And if you squeeze both the north, south, and the east-west walls together, then the whole grid gets a little bit smaller. And the amount by which it compresses is about 50% of the amount that the environment compressed. So the kind of conclusion from that is that although the grid cell has an intrinsic metric for space, generated by the rat's kind of internal self-motion cues, once a rat is familiar with an environment, the environment gets some control over that. So if you subtly modify the environment, that has an effect on the grid cells as well. So as with the head direction cells, they're integrating environmental and self-motion information. So we're now really seeing the kind of metric structure of this kind of spatial representation. And O'Keefe and the Mosers got the Nobel Prize, you probably know about this, for the place cells and the grid cells. Head direction cells are probably going to get one all on their own because they're pretty important as well. So what do grid cells tell us over and above the place cells? Well, they also show that there's metric information in the linear domain. They show that, again, the brain is forming these supramodal representations that integrate from different sensory modalities and also integrate different kinds of information. And they may be the way that the brain does trigonometry. So it may be that when a rat is trying to path integrate, let's say it's walking around in the dark and it walks two sides of a triangle, instead of computing with sines and cosines to get back to the start, maybe all it does is kind of use its grid cells to kind of work out where home is again. So maybe the brain has just made a kind of a lookup table for trigonometry or something like that. We really actually don't know what grid cells are for, if I'm honest. So we have to find out. That's the big task for next time. Okay, so there's one more kind of spatial cell that I should mention, and that's the border cells. So these are also mostly found in medial entorhinal cortex mixed in with the grid cells, but they've also been found in subiculum by Colin Lever and his colleagues. And these are cells that fire along the borders of the environment. Sometimes they fire at a distance from the border, but mostly they fire at the borders. And that may be one of the ways, for example, that grid cells know how to compress their fields when the environment compresses. So there are these kind of four spatial cell types. So I'm slightly running out of time, partly because of the lamp. So I should 5 more minutes. So, oh, that's okay. No, don't worry. So what I'm just going to do briefly is to introduce you to the notion that we can extend beyond the very simple environments that I've described to complex space. So we've talked about rats in very symmetrical small boxes, but the real world looks a lot more like this. At least it looks like this for us. So it's very, very kind of complicated. It's extended. It has multiple compartments instead of just one. And these compartments may be extended vertically as well as horizontally. So lots of animals, including ourselves, can move in all three kind of dimensions. So what happens in three-dimensional space? So we've started to look at what happens in multiple compartmented environments. And just to kind of briefly say that place cells don't really seem to care if there's more than one kind of compartment that the rat is in, that the rat can walk between, so long as they all look the same. So in an environment of identical small rooms, kind of like an office block, this kind of whole bunch of place cells all did more or less the same thing in every repeating unit of the environment. So even though the rat is walking backwards and forwards and has a lot of path integration information, the place cells don't care about that. They don't distinguish the compartments very much. We don't yet know if the rat itself knows or cares about these multiple compartments, and we didn't give it a reason to know or care. So all we can say at the moment is that latently speaking, place cells are not using path integration as a strong cue to lay down the places where their fields are. Grid cells, on the other hand, are slightly different. So if you put grid cells in a multiple compartmented space, initially they repeat their patterns in the two spaces, as if they think that the rat's just going back into the same compartment. But if the rat can do this over many days, then they start to form a more generalized representation where now the pattern flows out. And we've actually modeled this. So Francis Carpenter, who did this work, has modeled this and shown that the pattern is different in the two compartments and is best explained by a global map that's kind of covering both of those compartments. So grid cells have the capacity to use path integration to kind of differentiate the compartments. We don't yet know whether place cells can do this given sufficient information. We also know that that grid cells can do sort of what place cells can do in terms of using contextual cues to differentiate compartments. So they do this in a much more subtle way. So if you put the rat in one of these boxes that's different colors or different odors, grid cells, if you look between here and here, they very slightly shift their grids. It's a very, very subtle effect. So they're getting some information about context, but by and large, they're not particularly bothered. So we're kind of looking at multi-compartmented space. And I'm going to skip over this and just say that we're also looking at three-dimensional space. So the question for 3 dimensions is these kind of flat representations that I told you about, where grids kind of form this nice kind of grid, carpet of fields. If the rat could kind of float through 3-dimensional space, would we see something that looks more like this? So kind of firing fields that are stacked in a very organized way. And with head direction cells, is this ring attractor really a spherical attractor? So is there a head direction cell for every kind of direction in 3-dimensional space? And I'll just kind of give you the punchline there, which is to say that for rats, we don't think that's the case. We think that rats continue to use a ring attractor, even in 3-dimensional space, but I won't talk you through why we think that. Okay, so I'm just going to skip to the end here and give you the conclusions. So the overall conclusion, so what can the spatial system tell us about cognitive architectures? So the brain is clearly quasi-modular, so it's not one enormous deep network, but it may be lots of deep networks all doing slightly different things. So we've got part of the brain that does head direction signal, part of the brain that does the odometry, part of the brain that does landmarks, and so on and so on and so on. So we already knew that, but it's true for the spatial system too. It learns spontaneously, independently of goals. And it seems that the system is kind of driven to do this. So if you make some kind of change to the environment, then even if a rat is hungry and there's food there, the rat will kind of not care about the food until it's explored and checked out the change. So curiosity is a very, very important part of this system. So if you're making an artificial intelligent agent, you need some kind of exploration kind of capacity, some kind of curiosity. Some kind of motivation to constantly update. And I should say that even if a rat's in a very familiar environment doing a very familiar task, they never perform at 100% correct. And we think the reason is that sometimes they break away from the response that they believe is correct in order to test the other response, like go to the other arm on the T-maze or whatever, just to see if the parameters of the task have changed. So even in a familiar environment, to some extent, the system's always checking, might something have changed? So that's very important. The brain, we could say that it sort of uses symbols, so thinking of cognitive semiotics. So you could think of place and grid cells and head direction cells as kind of signifying different subcomponents of the spatial map. There are different encoding methods, so a lot of these architectures are feed-forward, some of them are recurrent. There's attractor dynamics in some of these systems, but maybe not in all of them, and so on. There's relational encoding, so we have distances between things. And we think that the brain does things to try and save itself effort. So I didn't talk you through the 3-dimensional stuff very much, but there's reason to think that the brain is trying to make a flat map of space in order to kind of minimize the unnecessary processing that it does, because using a flat map works pretty well even in 3 dimensions. And we could talk about that after if you want. And maybe the grid cells are a way of doing trigonometry and all that kind of stuff. So the brain kind of tries to find shortcuts, I think, wherever it can, to simplify problems in an adaptive kind of way. So I'll just put up my acknowledgements so you can see them, and I'd like to thank you for your attention. Sorry, I've kind of used up my time.
Thank you very much. We have time probably for one question, because one question already was here. One question, please. Alexei, please.
I think you missed the most interesting part of your presentation about 3D
I'm sorry.
Can you use this time to tell us?
I could, just very quickly, okay.
But you had too many.
Okay, so I'll start from here. very quickly because I know people want lunch. So grid cells, because of their metric properties, now for the first time we can ask kind of, what's happening to the representation in 3 dimensions. And so we started to kind of do this by trying to get rats to go in 3 dimensions. And the first kind of experiment was to record on a pegboard. So this is a kind of a climbing wall. And the question was, would we see a grid that looks like this on the wall, or would the kind of grid cell break down? And what we actually found is something entirely unexpected, which is that the grids form stripes on this wall. So, so the firing seems to be continuous in the vertical dimension, but periodic in the horizontal dimension. So it's as if the distance measuring capacity of the grid cell is following where the rat walks in horizontal space, but not where it walks in vertical space. And we also saw something similar in a different kind of apparatus where the rat was kind of walking up the spiral staircase. The grid cells fired periodically around every coil, but they fired continuously on the kind of different coils, as if they didn't know how high the rat had gone. So we started thinking maybe the intrinsic structure of grids is something like this. So the grid is kind of trying to provide odometric information for the horizontal plane, but it doesn't care about the vertical plane. And then recently we've been thinking maybe it's more than that. Maybe it's not just the horizontal plane, but it's the plane that the animal is walking on. So on the pegboard, the rat is oriented horizontally, so maybe that's why grid cells care about the horizontal plane. But if the rat could walk around vertically on a vertical wall, maybe we would now kind of see a grid. So we're looking at this, and the answer to that question turns out to be slightly complicated. So I can say very definitely that we don't see stripes anymore when the rat can walk around freely like this. We see blobs, but we don't see a grid. So we see these nice round fields. So they're not elongated. They have the same kind of dimension. They have the same kind of metric kind of properties this way as well as this way. But there's typically only 1 or 2 of them. We don't see any kind of regular kind of pattern. We're trying to figure out what's going on, and we think that what's happening is that the brain is not doing odometry very well. So the grid cells are struggling to kind of work out how far the rat has gone, and so they're dominated by the local cues, so they're dominated by the visual cues. So they're able to form a blob because there's enough visual information, but they're not able to space the blobs kind of sufficiently. So we think that the representation of vertical space is very degraded, which is why I said that I think the brain is trying to make a flat kind of map. So it's kind of more like this, really, except that these are kind of much larger than these. Sorry, does that answer the question? Sorry, I've taken up 5 minutes.
Thank you. Maybe one
Okay.
more question.
You can talk to the other night.
Okay, thank you.